This episode was recorded prior to January 20th.
Hello, and welcome to the ACR Journals on Air podcast. I am your host, Vicky Shanmugam. The workup of a patient with a positive ANA is the bread and butter of every rheumatology practice. But what causes a positive ANA? We know there are some genetic predispositions to autoimmunity, but we also know that some people develop autoantibodies with no overt signs of clinical disease. So can large-scale genome-wide association studies predict who is at risk for autoimmunity? The manuscript we will discuss today is Genetic Analysis of Asymptomatic Anti-Nuclear Antibody Production. It was published in Arthritis and Rheumatology, and a link to the manuscript and disclosures are in our show notes from today. We're joined today by the lead author, Dr. Mehmet Hocaoğlu. Dr. Hocaoğlu was born and raised in Turkey. He studied medicine at Istanbul University School of Medicine and became interested in immunology when he was a research assistant studying B and T cell co-stimulation pathways in myasthenia gravis. After graduation, he trained as a research fellow at Mayo Clinic under the mentorship of Dr. Duarte Garcia and Dr. Crowson, using the Lupus Midwest Network, or LUMEN registry, a CDC-supported national lupus registry, to investigate the incidence, prevalence, healthcare utilization, and comorbidities of lupus spectrum disorders. Mehmet then completed training in internal medicine at the University of Maryland, and under the mentorship of Dr. Hong, he studied genetic epidemiology of rheumatic conditions in biobanks around the globe, including the UK Biobank and FINGEN. He is now a second-year rheumatology fellow at the University of Pittsburgh and is fortunate to work closely with the senior author of this paper, Dr. Amar Sawala. Welcome to the show, Mehmet.
Thanks for having me, Vicky.
So, Mehmet, I wonder whether you could provide some background to this study. Positive anti-nuclear antibodies are very common. Why was it important to do this analysis?
Absolutely. Well, as you said, ANA positivity is quite common in the population. Population-based studies in the US actually estimate that around 14% of the general population is positive for ANA. Obviously, as rheumatologists, we know that these numbers change with the respect of the titer and the pattern of the ANA, but overall, about 14% of the general population is actually positive. And we also know that the ANA positivity has a strong association with several autoimmune diseases such as lupus, scleroderma, and others. But we also know that the vast majority of individuals with positive ANA in the population are asymptomatic. There's actually a seminal paper in New England Journal of Medicine back in 2003, which really inspired us, that showed that the ANA develops several years before the clinical onset of lupus, and several other autoantibodies in lupus, they were shown to be gradually accumulate till the symptom onset. This really suggests that the breakdown of autoimmunity happens in phases and the clinical disease develops after a certain threshold is passed. Hence, individuals with asymptomatic ANA positivity might be in that intermediate state, if you will, in the progression to autoimmunity. And we also know that both genetic and environmental factors play an important role in this progression. And through the years of genome-wide association study efforts, we got a good understanding of what are the common genetic risk variants that play a role in development of lupus and other autoimmune conditions. But a gap in our understanding was we know what are the common genetic risk variants that are associated with lupus, but we did not really have a very deep understanding of what are the genetic variants that determine that initial progression, if any, of developing this asymptomatic phase of the disease where you develop the autoantibodies, and how are the people who go on to develop lupus or other autoimmune diseases and who have positive ANA are different than the people who have positive ANA and never go on to develop the disease.
It's what makes them special. It's really the most important question.
In that line, in this study, we try to explore what is the genetic architecture of individuals who have ANA positivity and never develop symptoms, and then individuals who do not develop ANA as well as individuals who develop autoimmune diseases after starting as positive ANA.
So tell our listeners a little bit about the All of Us data set. This is an NIH-funded program. Tell us who is enrolled, why do they choose to enroll, and what is being collected on those individuals?
Yeah, great question. I'm actually very excited to speak about that because I think it's an amazing resource. The All of Us research program is an ongoing biobanking effort aiming to enroll about 1 million individuals across the United States. And the goal is to gather lifestyle surveys, genomic data, as well as other omics data set, looking at how proteins and the methylation of other kind of biological markers are in these individuals, and link them with their electronic medical records so that we understand what types of illnesses they have. And as of latest data release, there are about 300,000 individuals who have both genetic and electronic medical records available. But this number will likely grow in the next few years as there is ongoing data collection and curation. And I think a lot of people enroll in this because I think this is a great opportunity to really study in depth what are the genetic and environmental determinants of common diseases, for us, such as lupus, autoimmunity, but I think all of the common diseases that are out there, such as diabetes and heart disease and others.
And tell us a bit about how researchers and others can apply to use data from the All of Us registry. It's pretty unique for a rheumatology fellow to be allowed to use a database this large, but that is part of the mission of the All of Us registry, to really bring research to communities affected and to help anyone who has an important research question be able to use the registry to answer some of those questions. So what steps did you need to take to conduct this study? And could anyone listening to this podcast who has a burning research question follow in your footsteps?
Yes. So I think one of the things that make All of Us a fascinating research resource is the fact that it's quite easy to gain access to the data. I personally actually have experience working with other biobanks across the world, and I can confirm that All of Us has the easiest and the fastest application process. And I think they did an amazing job to make it so available and so easy to access for researchers across the country and even across the world. Now, the main limitation is that researchers must be affiliated with one of the registered institutions with All of Us, but this already includes many universities and academic medical centers in the US. Then all the researcher have to do is to go through an identity verification process, and then there is some training to understand what All of Us is and the regulations are. And then after these are completed, you can get access to the data actually in just a few days, which is quite amazing. I mean, these process for other biobanks across the world can take everywhere from 6 months to a year, but in All of Us, you can actually establish your access within just a few days, in a week. So that's, I think, a really big strength of the All of Us data set. Now, one other restriction is that all the data has to be analyzed in their cloud environment, cloud computing program, because of the sensitivity of the data there. But also All of Us has amazing resources that explains the details of the data, as well as many tutorials to help researchers advance their data analysis skills. So yes, I think both new and experienced researchers with a burning research question should explore this amazing source. I specifically want to highlight that if you're interested in gene-environment interactions, I think All of Us is a great resource because you have both the environmental surveys and then the genetic data, as well as the electronic health records. So a lot of research could be conducted in that avenue. And also, as you pointed out, I think this is a quite innovative approach that, so to speak, bring researchers to the data, where everybody could ask questions and analyze the data from their perspective. And this really enables studies such as the one that we conducted to be performed with really limited financial and other resources, because the front-loaded investment has already been done by collecting and curating the data, and all you have to do is come up with questions and analyze it in the way that you are interested. And I'm actually really excited about the possibilities of such a model would offer for enabling research by early career researchers such as me and others, as well as research into rare diseases, which is not really easy to get enough funding and resources invested to collect the necessary amount of data. And I'm also excited. I mean, this is not only all of us. We know that there are similar biobanking efforts actually going on in every part of the world. There are, dozens of biobanks that are being collected right now. And I'm also for the future very excited that, eventually we can get to a point where we can achieve very large sample size to finally benefit from one of these, advanced machine learning models to better make sense of the biological data, which otherwise would not ever be possible because of the big investment needs for an individual funding agency or even individual country. So I think it's a very exciting model that we will continue to see, good research coming out of it.
Yeah, I totally agree, and really thrilled that you did this research and published it so that others can see how you did it. So let's talk a little bit about the study design. Which participants that were enrolled in All of Us did you select for this analysis, and how did you design this study?
Yes, so we selected individuals who had either positive or negative ANA test results in their medical records in All of Us, and excluded those who had any diagnostic code or prescriptions suggesting autoimmune disease. And we have designed the study as a classic genome-wide association study where the individuals who are asymptomatic from an autoimmunity standpoint and have a positive ANA defined as cases, so to speak, and the individuals who are asymptomatic and have a negative ANA test defined as controls.
And how did you determine the ANA status? Tell us about the access to the electronic medical records through All of Us, and how much detail do you get about the labs? How does this all work?
Absolutely. Well, for the participants of All of Us, when they enroll into this study, they give consent for their medical records to be released to the data set. Unfortunately, because of the way that the US health system is set up, this is not a very easy task because individual might have electronic medical records in different health systems, which may use different versions of electronic medical records. So there is a centralized process where for each individual, all of their electronic medical record data is gotten together and then harmonized, and a data set where kind of the harmonized data from all the labs and all the diagnostic codes and prescriptions released to the researchers. So what we get as All of Us researchers is not the underlying medical record per se, but that harmonized version of the medical records released by the All of Us research consortium. And that has the lab results, the lab comments, the titers. Now, there are some missing information for some certain aspects of the electronic medical record, as we see in all of these kind of harmonization process. But we were able to get the ANA lab status for individuals in All of Us. And because of the potential kind of missingness with the titer or different parts of the electronic medical record, we actually defined ANA positivity as reported positive by the performing lab. And if the titer information was available, then we only included ones that had a titer greater than 80.
So just to be clear, and I think our listeners are going to be very interested in this, you were looking only for people who'd had immunofluorescent ANA testing. So you were not including people with like a positive Bioplex or ELISA test. Is that correct?
Yes, true. And thanks for kind of bringing that point to clarify. And we did only look at individuals who had a immunofluorescence-based testing of ANA because that is kind of what we take as gold standard and commonly use in our clinical practice. But I think the good news was that actually the majority of the individuals in our data set actually had their result per immunofluorescence. So we didn't end up excluding a lot of patients, but we did restrict it for immunofluorescence ANA test only.
And am I understanding correctly, these people were not because they were enrolled in All of Us getting ANA testing. These people were just enrolled in All of Us and happened to have an ANA result in their routine medical records, which means they must have had some reason for somebody to send it in the first place, correct?
Exactly, yes. So this is not a seroprevalence, so to speak. These people were tested as part of their routine clinical care, and we established this cohort from these people who had their ANA tested through their routine care. If we had a research-based testing of ANA at the end of All of Us cohort, that would have been truly amazing, but unfortunately that is not available.
Yeah, no, I get it. And so it's really a convenience sample of people who had these results done through routine clinical care. How did you assess whether or not people had an underlying autoimmune disease, and particularly whether or not they had underlying lupus, say, or scleroderma?
We used the electronic health record data, as I said, and our goal was to have a very high sensitivity to exclude any autoimmune disease from our cohort, because I think the interesting part of this study was to look at the genetic architecture of people who have positive ANA and not have any autoimmune disease, because we already know that people are enriched for certain genetic risk loci when they have autoimmune disease. So when you look and find autoimmune risk loci for people who have positive ANA and autoimmune disease, that is not an unexpected finding, so to speak. So our goal was to make sure that, as to best of our ability, that all of the individuals in our cohort did not have any evidence of autoimmune disease. So the way that we achieved this is that we have looked at 30 or so. I don't know the exact number out of my head right now, but we have looked at every single disease who is known to have an autoimmune origin, and then we excluded even if they had a single code of that disease in their medical record. The full list is in our supplementary table, but we did really kind of not only lupus and scleroderma, but we excluded patients with sarcoidosis, patients with IBD, patients with vasculitis. So we did not really restrict this restriction to lupus, but we have defined a really broad definition of autoimmunity. And our goal was to, as I said, have a very high sensitivity to identify anyone who has any sort of autoimmune disease so that we can exclude it from our cohort. And as an additional safety check, so to speak, we also excluded anyone if they had a prescription for a immunosuppressive medication in their medical record, because sometimes because of the missing data, they might not have any codes. But if we saw that even if they don't have a code for an autoimmune disease, they have a prescription for, let's say, Plaquenil or hydroxychloroquine, then we ended up excluding those patients as well, because then we're worried that they may have an autoimmune disease that is not captured from the electronic health record harmonization process. So that's kind of what we did. And about the lupus specifically, that was actually part of our second analysis part for lupus. We did use it as a comparison for our polygenic risk score analysis. And for that, our goal was to have a high specificity to capture real lupus cases. So for that specifically, we basically required that you had at least 2 codes of systemic lupus erythematosus related ICD codes, as well as a prescription for one of the medications commonly used in lupus, such as belimumab or hydroxychloroquine or others.
So I wanted to just go over that pretty carefully, because there were really 3 cohorts of patients that you studied. A group of patients who had no clinical disease but were ANA positive, a group who had no clinical disease but were ANA negative, and then a group of subjects who carried a diagnosis of lupus. Why did you include that lupus group?
Great question. And thanks for asking to give me a chance to clarify. So the lupus group was not involved in the initial GWAS analysis. In the initial GWAS analysis, what we were interested in looking at was the genetic architecture difference between individuals who have positive ANA but no evidence of clinical disease, and compare them with individuals who have negative ANA and no evidence of clinical autoimmune disease. And we did this initial analysis and we got our results. As a second step, we wanted to look at the same problem from a different angle. Now, through years of GWAS efforts, we've been able to identify many lupus genetic risk loci. There are somewhere between 200, 300 genetic risk loci that have been identified for lupus. And in using them and simply adding up the genetic risk of lupus, we can construct a score for each individual. And this score tells you how much genetic risk of lupus that you have. And as expectedly, individuals who have lupus tend to have more genetic risk for lupus compared to individuals who do not have lupus. But this is not an absolute thing that if you have a very high genetic risk of lupus, it's guaranteed that you're going to develop the disease. That is not the case. A lot of people who have very high risks of genetic risk for lupus do not end up developing the disease. But this is a way to kind of look at it from a genetic angle and see how much genetic risk of lupus you have. And as a second point, we said that, well, we ran the classical GWAS analysis within the cohort of our asymptomatic ANA positive individuals. As a second step, can we construct a lupus genetic risk score for them and compare if they're on average different in their genetic risk score or genetic risk burden for lupus? And when we ran that analysis, obviously it's a good idea to include lupus patients as a positive control, so to speak, because there are actually many different ways that you can construct such a genetic risk score. And if you construct a genetic risk score and you find that the lupus patients are expectedly at a higher genetic risk compared to your cohort, then you have, so to speak, some sort of an idea that, okay, my approach is working and I was actually accurately capture the higher genetic risk in lupus patients. So that is the reason why we included the lupus group, so to speak, as a positive control for that last stage of polygenic risk score analysis.
And I wondered, can you walk us through a bit more about the analysis? Because GWAS obviously involves lots of complex statistics, and I wondered whether you can simplify some of the steps for us. As a researcher, after you got all the necessary approvals from all of us to access the genetic data, you needed to do some quality control measures to set up the analysis algorithm. Would you mind just explaining that process for us, and why is it essential to map the reference populations you're using with the 1,000 Genomes Project? And why does that process then cause you to exclude some specific populations from the analysis?
Sure. Well, whenever we undertake a GWAS, as you said, it is very crucial that we go through the necessary quality control steps on our data. And as you also said, unfortunately, quality control in GWAS includes, many steps, and it is actually a little bit complicated, but I can do my best to kind of give the general idea of what we're doing in these quality control steps. When you look at it from a generalized view, there are two big ways of doing quality controls. One is that we looked at what we call variant level quality controls, making sure that the genetic variants that we're including in our GWAS are high quality variants. And then the sample level quality controls, meaning that the samples that we took from individuals to process their genetic data were actually high quality samples. And the ways to look at this problem is, one, basically we looked at if there are a lot of missing data from either the variant side or the sample size. If there are actually a lot of missing data for whatever reason, these variants or the samples were not processed, that makes us worry that there was something wrong during the processing. So we exclude those variants and samples from our analysis. Then there are actually ways of looking how much expected genetic allele frequencies you're expecting to have in the population. And if there's a lot of deviation from those expected counts, then we also exclude those variants from the analysis. And then we also exclude very rare variants, less than 1% in the population, both from a statistical standpoint that it is harder to get the necessary power to adequately analyze those variants. And those are kind of the big picture quality controls that we do. But another very important point, as you have pointed out, with the mapping the population to our reference population, such as the 1,000 genomes, is because when we run a GWAS, basically what we do is we compare the allele frequencies of certain genetic variants between cases and controls. And it's actually quite simple. You basically look at a genetic variant X at a certain position in the genome, and you look that it's 30% in the cases and then 20% in the controls. That tells you that this genetic risk variant is enriched in the cases compared to the controls. But in GWAS, an important point of caution that you need to have is a phenomenon called population stratification. And what that means is that a lot of the genetic risk variants and their allele frequencies differ significantly between different populations that are coming from different genetic ancestries. So when you run a GWAS without correcting for that difference from their own genetic population, then you can find hundreds, if not thousands, of differing allele frequencies in these genetic variants. So we do have to get an idea for each individual that we include in our GWAS, what type of genetic population are they coming from, and then separating them into different genetic populations, analyzing them separately, and in the end, meta-analyze the whole results. And that kind of corrects for those differences that you have in your allele frequencies between different genetic populations. And the one way to achieve that is, as we did, you basically compare your cohort with a reference cohort, such as the 1,000 Genomes Project. And maybe everybody is not aware that 1,000 Genomes Project is a whole genome sequencing effort for about 3,000 individuals. That was now then, I think, more than 10 years ago. And it's just individuals who are from different genetic ancestries that are got together and they underwent genetic analysis. So we know, what type of genetic ancestry they're coming from. So we basically look how much different or similar our cohort is to this reference population. And through that, we get a sense of what genetic populations are available in our cohort so that we can separate and analyze them in individual GWASs and then combine them in the end together. And when we did that analysis, we actually found out that individuals from South Asian-like genetic ancestry and East Asian-like genetic ancestry, there were not actually a lot of individuals from those genetic populations in our cohort. So we had to exclude them from our analysis because we just didn't have enough numbers to be able to reliably run our GWAS analysis for those individual genetic populations.
Okay, so now that you've explained how you set up the analysis, can you tell us a little bit more about the groups that you did analyze? And I guess maybe talk us through table 1. What was the breakdown of your demographics?
Absolutely. So in table 1, we have about a total of around 2,000 individuals who were asymptomatic and had ANA positive tests in their medical records, and then about 3,600 individuals who were asymptomatic and were ANA negative. And majority of the individuals in our study were from European-like genetic populations, but we also had a significant number of individuals who had African-like or mixed American-like genetic populations. Just to say that these are just genetic terms that are from these 1,000 genome reference populations, so they don't really correspond with ethnicities of the individuals. But basically, the mean age of the cohort was between 50s and 60s throughout different genetic populations, and the most individuals were female. And when we compared the demographics between cases and controls, we did not really see any statistically significant differences with respect to age or sex.
Okay, so the fun graphic, though, is figure 1, which is your Manhattan plot. And since this podcast is audio only, I'm wondering if you can describe it for our listeners and then just walk us through how we should interpret it.
Yes, no, after going through this painfully constructed analysis process, we get the figure 1 in the end, which, as you said, is kind of just one figure that summarizes everything that the GWAS is showing to us. It is actually quite simple to understand. The x-axis of this figure basically depicts the genetic variants across the genome and their location. So basically, you sort all these genetic variants that you have compared in your GWAS depending on their position and their chromosome. So if you have, let's say that I have one genetic variant in chromosome 1 at position 100 and another one in chromosome 1 at position 200, I basically, individually depict them in order, and you can see the chromosomes depicted in the x-axis. And the y-axis is basically the p-value for the individual genetic variant that was included in the GWAS. So if you have a genetic variant that has a very strong association, meaning that they have a very small p-value, you would see that depicted really top on that y-axis. And there are two thresholds in that Manhattan plot. One is the red dashed line, which is just a threshold for genome-wide significance. And that is actually quite a low p-value, 5 to the 10 to the minus 8. And the reason that we require such a small p-value to consider something statistically significant is because when we run a GWAS, we kind of run millions of variants throughout the genome. So just by chance, we can expect to see many differences between cases and controls. And that threshold kind of corresponds to a 5% p-value threshold, which is more traditional in other areas of research, corrected with the massive amounts of tests that were running in parallel in a classic GWAS study. And there's also another threshold that the readers can see some other time when they look at it, is that is the blue threshold there. And that is just the genome-wide suggestive significance level. And that is for the variants that are above that threshold. We're not really confident that this association is statistically very significant, but it's just a lower threshold for us to increase our sensitivity a little bit and see if there are any weaker signals that we can detect in the GWAS. So that's kind of how you look at a Manhattan plot.
Okay, so for us members of the audience who are not geneticists, including myself, if there are none of the blue or orange dots above that red line, it really means there are no genome-wide significant associations. Is that correct?
Yes, you're absolutely right. If you don't see any dots above that red line threshold, then that means that there are no genome-wide significant associations. And we were actually quite surprised to see this initially, because when we ran our analysis, our hypothesis at least was that we are going to identify some limited, at least, genetic overlap between individuals who are asymptomatic and have a positive ANA versus individuals who are ANA negative and don't have also any autoimmune disease. But this did not end up being the case. We could not detect actually any genome-wide significant variant between ANA positive individuals and individuals who have negative ANA and no autoimmune disease. But actually looking back, this corresponds with a study that was done by the NIH a few years ago. There is the National Health and Nutrition Examination Survey that the NIH does every certain years throughout the US to get an idea about kind of the environmental risk factors and the disease outcomes throughout the US. And they had samples available from each cycle of the survey. And NIH researchers looked at the prevalence of ANA in the US populations through that sample. And they have actually found that there was a significant rise in the prevalence of ANA for all genetic populations in the US. And that happened within the last 20 years or so. And there was about a 50% rise in the prevalence. Something like that actually very hard to explain for a trait that is under strong genetic influence, because if something is under strong genetic influence, it takes many generations before you see such a difference in the prevalence. So I think this study that we did gives a nice genetic point view of the same result, that the effects are largely environmental for ANA positivity in the population.
Yeah, really, really important data. And so even though it looks, quote, negative, it's really telling us something crucial. So I'm so glad that you pursued this. So when you started digging deeper and looked at the associations in the individuals who had clinical lupus based on their EMR records, did you find anything interesting when you compared those individuals to these asymptomatic positive ANA individuals?
Yes, and thanks for kind of bringing that point up. Is that, as I said, our main goal for including the lupus group was to have a positive control for our polygenic risk score analysis. And this, I think, ties up nicely with our GWAS results, is that when you calculate an individual's polygenic risk score or genetic susceptibility to lupus, and you compare the genetic susceptibility to lupus between individuals who are asymptomatic and have positive ANA versus individuals who are asymptomatic and have a negative ANA. So when you compare that two groups, you don't really see any difference with respect to their genetic lupus risk. And I think that is very interesting. I think that also kind of reinforces what we have seen in the GWAS, that kind of negative finding. But I think there are some implications to our clinical care. I mean, as a rheumatology fellow, similar to other rheumatology fellows or rheumatologists across the country, we see a lot of individuals who have positive ANA but don't really have any symptoms of lupus or other autoimmune disease. And it's funny because I used to say that, well, because they ask what they means, and it is really anxiety provoking to them because they read about lupus, they got concerned about the significance of this test, and they used to ask me, what does that mean? And I used to very crudely say, well, you may have the gene for lupus, so to speak, but you may never end up developing the disease, that this is not something to worry about. But in light of these actual results, that is actually not correct. Individuals who have a positive ANA and don't really develop any symptoms, even genetically, they are on average, obviously, not at higher risk for lupus compared to individuals who are ANA negative. And as I said, this is a statement for the population on average, not to the individual patient. But I think that this helps in our discussions with our patients when they come to clinic and they ask what this means, what this test means for lupus. I think this genetic evidence gives me, at least me personally, more confidence to tell them, well, this is something more that I can tell you to be reassured about, is that even genetically, people who develop this positive ANA antibodies in the population on average are not really at higher genetic risk for lupus. So I think that is very helpful for the clinical care as well.
Yeah. And I feel like that's a incredibly important advance. And, I'm so thrilled that you did this study. So walk us through some of the limitations of this type of analysis and tell us a little bit about kind of where this needs to go from here.
Absolutely. Well, I think the most important limitation of our study is really inherent to the definition of ANA itself. ANA is an umbrella term for numerous potential antibodies, and we already know that the clinical significance of ANA changes per titer or pattern, so that when we say, that we're analyzing a positive ANA, that is kind of a positive ANA that we see in the population on average. And we know that different titers and different patterns can have different genetic architectures because we also know that they have different clinical significance. So we did actually do a sensitivity analysis by restricting our analysis to individuals who had a titer greater than 160, which we know that has a stronger association with autoimmune diseases. And our results really didn't change when we restricted the analysis to individuals who had a higher titer. But then again, whenever you do a sensitivity analysis, you do lose some power. So the sample size was limited for that subgroup analysis. So I think because of this, our conclusions about the genetics of asymptomatic ANA positivity should be viewed as on average that we observe in the population, not for all possible titers or patterns of ANA. And the other thing is, obviously, this was a convenience sample, as you put. So this is a cohort established through electronic health records. So missing data and misclassification biases are always possible. But I think for the purposes of our study, we try to really mitigate this by having a really broad definition of autoimmunity, including both medical codes as well as prescription data. And because our goal was to rule out autoimmune disease, not to specifically identify high confidence cases, I think this is a little bit of a less concern for the purpose of our analysis, because all we wanted to do was to find autoimmune cases to exclude them from our analysis, not to kind of find them in a high confidence way. But then again, as I said, this is a limitation. And finally, one thing that I'm also excited about for future research is that all these analyses that we did do not really assume gene-environment interactions. So could there be a gene-environment interaction that play a role in the development of asymptomatic ANA? That's certainly possible. Our methods do not really assume gene-environment interactions. And I think that is an area of active research for the future.
Yeah, and I think there have been a number of people saying that for a while, that we can't study genes and environmental triggers independently. And if we want to understand autoimmunity across the lifespan, we need to be able to analyze that intersection of environmental factors with genetics. And that's going to require a slightly different way of doing research.
Absolutely, yeah.
So I'm going to change direction now because I've always loved hearing about how people get their start in rheumatology and in research. Tell us how you got bitten by the research bug and what attracted you to immunology.
I had a fascination basically with the immune system since early medical school. I have specialized research interests, but I always been kind of a generalist in my heart. And I was initially drawn to immunology because of its kind of overarching role in medicine, from autoimmunity to cancer to infection to transplant. So it was this one system that kind of seemingly unify all these different areas of medicine. So that kind of led me spending some time at an immunology lab in our medical school. And as my interests grew and I studied more and learned more about the immune system, I was really hooked. And at that point, I actually decided to pursue a fellowship in rheumatology even as early on, because that was the specialty that was going to allow me to deal with the immune system the most. And, that's kind of how I ended up with rheumatology and research in the beginning.
So you had quite a journey from Turkey to the Mayo Clinic, then the University of Maryland, and finally to Pittsburgh. Tell us how you ended up at the Mayo Clinic, and do you have any advice for other international medical graduates trying to find a spot in the US?
Yes, well, it was a very long and very rewarding journey. The way that I initially ended up in Mayo Clinic was that my mentors back in my medical school had previous research collaborations with faculty there. So they recommend me as a postdoctoral research trainee to them. And this actually started my journey as a researcher in the US. So I was kind of lucky to have that support to, push me through my research career. Regarding international medical graduates, I am one, and it's obviously a long process with, many examinations, clinical rotations to get the necessary experience. And I think IMGs who are looking for a position in the US come from diverse backgrounds and diverse interests. So I think it's the best idea to talk to someone who is sharing your background and sharing your future interests. But me specifically, for someone who is really interested in doing research, I can advise that maybe coming to the US as a researcher first and doing, research for a few years and then pursuing clinical training is really a good idea. Because if you're really interested and serious about a research career, you will have to stop and do a few years of dedicated research at some point of your career. And I think doing that in the beginning, getting the research skills, under your belt, and then transitioning to the clinical training might be a very good way of achieving this.
Yeah, I think that's very good advice. And you, as someone who has obviously successfully combined your research interests with your clinical training, how do you think you managed to pull that off? And do you have advice for others as they start out in their career on how to maintain a bit of a research focus while juggling clinical rotations?
Yes, well, I mean, it is definitely not easy, but I think the most important part of it is being persistent and having the right expectations and timeline for your research interest. I think from the get-go, as clinical trainees, we have to understand that our experience and timeline is going to differ from someone who is doing dedicated research, such as a PhD research trainee or a postdoctoral research fellow. But I think that as long as you're proactive and have long-term planning about your research trajectory, a lot could be achieved even during your clinical training. And I think identifying a research mentor in your institution as early as possible is key. I was personally very lucky to be introduced to different research mentors by my earlier mentors, so to speak, in a chain reaction throughout my research experience. But I think it's always a good idea, if that's not available to you, to be very proactive, email and meet potential research mentors as early as possible, in your clinical training. And the research labs are generally very receptive to new trainees. They are very accommodating to clinical trainees specifically because they understand how much clinical load we carry. So I think that is the key to kind of talk with many different people and find the best fit for your future goals and your research interest. And then once you kind of find a mentor, which is the tough part, so to speak, then you have to pick up a project where you can learn. And I think this is probably not a universal advice, but probably, finding a project that you can make significant progress, even complete in your clinical training might be very important, because when you start a project as if you're going to complete it, even if that's not going to end up being the case, that really pushes you to push hard and, find the necessary accommodations to achieve this. But in the end of the day, it is going to be a very tough process. But because I'm a clinical trainee, I always, try to find analogies between clinical training and research training. I think it's quite similar that when you first start a project and when you first start doing research, it is going to be very tough. The concepts will be, tough to understand. The analysis will be tough. Experiments will be tough. Everything will be tough. But as the time moves on, I do think that as long as you're persistent, you start to get more fun out of it. Basically, it is very analogous to you starting as an intern. That is probably the hardest part of the clinical training, but that is not representative of your experience as a physician in the future. It's an outlier. So those first year of research or first 2 years of research is going to be very hard, but that is not going to be representative of your experience in the future. It's going to be the outlier in a very similar way. So that's why you have to kind of push through and be motivated about the future. And eventually, I think a balance could be found where you can do as much as you can get through your clinical training.
Oh, that's really good advice. And I appreciate you sharing that kind of perspective on taking the long view through the ups and downs of the challenges of research. So, Mehmet, I really want to thank you for sharing your research with us today and your experience as an IMG building your research career. Thanks for joining us.
Absolutely. Well, thank you for inviting me and your interest in our research perk. It was my pleasure to, talk about this research as well as my experience as an early career researcher. And I want to thank my mentor, Dr. Amr Sawalha, who supervises research, and Dr. Casares Marfil, who helped me with the analysis and the manuscript preparation, and was a co-author in this research.
Wonderful. And today I also give a hearty thanks to everyone enrolled in the All of Us registry. Enrolling in research studies is a huge commitment, but this collective effort really helps us accelerate health research and bring medical breakthroughs to improve healthcare for all of us. Thank you all for listening to this podcast. ACR Journals on Air is a production of the American College of Rheumatology. Please follow at AC Rheum on social media. Email us at podcast@rheumatology.org and rate and subscribe to our podcast at acrjournalsonair.org or wherever you get your podcasts.